Hi guys, I'm Ria Shah and I'm here to talk about the most enthralling topic in biology, which is gene expression and transcription factors. So we're all familiar with DNA. It's the molecule inside you that contains genetic information, making everyone their own individual. This could include the place where you get your brown eyes from, or even the ability of your tongue to fold. Because believe it or not, that's something people look for in genetics. But let's dig a little deeper into DNA. Now I have a DNA molecule right here, and it's made up of ATCG nucleotides. And remember, ATCG stands for adenine, thymine, cytosine, and guanine. And they've rearranged themselves into different sequences within DNA, and there's multiple segments of that. So those segments could all represent different genes. So if I circle this segment here, that could be the sequencing of a protein that makes up the gene for your eye color. Now this is where the transcription factors come into play. Think of these proteins as a computer code. They come in looking for a specific sequence of DNA that they are programmed to detect, and with that they bind to the DNA in order to transcribe the genes. The transcription of genes can also be called gene expression, and this is when a gene from DNA is essentially activated in order to make mRNA and proteins. So, going back to the idea that the segment of DNA was the eye color gene, let's say for green eyes, the transcription factor will bind to the sequence, as you can see here, and then it'll transcribe the DNA into messenger RNA. And that mRNA will allow for the gene to make a protein. In this case, the protein will be melanin that contains a green pigment in order to produce green eyes. Now, how is gene expression measured? That's my favorite part of all. Today, we have a modern technology called DNA sequencing. I'll walk you through the process. So, a chunk of DNA is first extracted from a cell. The DNA is then transcribed into RNA because the RNA contains the coded region which we can isolate for use. The RNA is then reverse transcribed back into DNA because DNA is what the machine will read. The fragments of DNA are copied thousands of times and placed standing upright on a board, and they're ready to be scanned. The scanner, or the sequencing machine, will then use a laser to go through the different layers of the DNA and take 4 different pictures. The 4 different pictures all correlate to the ATCG molecules. Then the data is put into a computer and computational analysis is done in order to determine the ATCG sequence and which genes got expressed. Gene expression can help scientists make new, vital scientific breakthroughs. I hope this helps you learn a little bit more about gene expression and transcription factors. Thank you.